Okay, today I want to talk about one of my favorite topics, environmental toxicology. And it's a slightly longer slide package than normal. So I may go a little bit too long today. If you have to walk out, that's fine. I want to just keep everything in the video though. So I think like maybe just 3 or 4 minutes long. But today we'll talk a little bit about general toxicology. And then I'm going to use the example of heavy metals. So last time when we were talking about groundwater, I used the example of organic contaminants to talk about remediation and dispersal. We can talk about organics today, but I think heavy metals are interesting to illustrate some aspects. We'll talk about detoxification mechanisms, and then we'll talk about sort of heavy metal sources in the environments and ecological effects. And then we'll come back after break, and then we'll talk more about heavy metals in freshwater and nearshore environments, and then we'll move into our topic where we talk about water treatment. So just some general comments about pollutant chemicals and their toxicity. They can come in various forms. You can read about this in the text. You can categorize things in different ways, organic, inorganic, radioactive, biological, and these are useful at some level. But there are some kinds of materials that have multiples of these, like certain radioactive materials. For instance, thorium is oftentimes more toxic because of its heavy metal characteristic than the radioactivity that it is imparting, depending on the isotope, of course. And we also have chemicals, which we'll talk about today, which are somewhere as a cross between organic and inorganic, what we call organometallic compounds. So, these kind of categories they're okay, but they're not great. Another thing we want to think about is not all chemicals are toxic in the same form, right? Sometimes they come in a liquid or a solid or powder, a vapor, an aerosol, aqueous, and some contaminants, mercury is a good example, can be found in all of these various forms, is not as toxic in each one of those forms. So that's something we have to think about. And this is kind of a flow chart of what happens, during the assessment of an environmental contamination. The first thing, which we've already been talking about a lot during the semesters, is kind of fate and transport. How did something get into the environment? Where's it gone, come from? Where's it going to? Is it dispersing? Is it not, etc.? Is it soluble in water? And then the next part of this is sort of determining that experimental effect. This is what's going to decide if it's just a contaminant or a pollutant. And there's a lot of gray area between here. And the EPA, for instance, has this category of chemicals it calls contaminants of concern. Things that have not quite reached the level of having an official level that defines it as a pollutant, below which things need to be kept, but they're kind of keeping a watchful eye on it. Things can be on that list for a really long time. Doesn't mean that they don't have detrimental effects. It just means that the process through which that is codified in the US hasn't been completed. And then the next thing is to think about impacts on target populations. We'll talk a little bit about this more today, but not all members of all populations have the same amount of sensitivity. In food webs, there may be different parts of the web that are more or less susceptible to that contaminant. And this, of course, depends a lot on how toxic a material is. So we want to talk about a rating scale that we have that defines toxicity. And then, once all this is done, that's when you start to devise your remediation methods. So another important thing to think about is, whether we're talking about humans, I'm going to use the example of humans, but you could think about other organisms as well. What are the exposure pathways to contaminants? And so for humans and other mammals, we really have these kind of 3 main pathways through which materials can come into the body: ingestion, inhalation, and dermal contact. And again, some chemicals can be toxic through all those pathways or only one of those pathways, depending on the concentration, et cetera, et cetera. And so when we start to evaluate an environmental contamination situation, we want to think about how people might likely be exposed. Are they going to get it out of the air? Are they going to get it by touching contaminated soil? Are they going to get it by bathing in water that has it in the water, et cetera, et cetera. So this is really critical that when toxicity is determined for different chemicals, they are determined through multiple different pathways. So that when we give the quantification of what the amount of toxic material is, something that we'll talk about in a moment as an LD, we'll have different LD values. This is the kind of maximum toxicity lethal dose depending on these pathways. And there are other pathways. If you were in a military scenario, there are other, for instance, if you were had shrapnel and it went into your body, that would become intramuscular. That's not so common for the civilian environment, so it's not included on that diagram. This is a toxicity risk assessment conceptual model for a project I happened to be involved with here for the US Army about 15 years ago, involving uranium contamination, sort of some sites here in the islands. And this is just this is a snapshot of it, but we've got contaminated soil. And there's, in terms of that fate and transport and where stuff is going, we can have water kind of infiltrating and percolating down and carrying some of the contamination down with it. We can have surface runoff bringing particulates. We can have particulate and gas emissions, especially very fine dust particles to which our heavy metal might be adhering to get blown up into the atmosphere, or in this particular case, because some of the contamination was on a firing range where they were firing stuff that was sending plumes of stuff up into the atmosphere. And then you can ask, well, what are the mechanisms of transport? Groundwater, surface water, particulates, plant uptake, air. These are things that, in addition to, you can have things living on the soil, for instance, so people playing in the soil, kids. And from each of these pathways, you can say, well, how might a person come in contact with that? Are they going to touch it? Are they going to breathe it? They're going to get it on their skin? They're going to ingest it? And so for each one of these pathways, from each of these sources, from one contaminated environment, you need to figure out not only what are the lethal doses and concentrations for both chronic and acute exposure, but what's the likelihood of the average person in these environments for different amounts of times to get each one of these pathways. And that's what comes into this health risk assessment. So that ideally you can say, yes, even though there's contamination here, the risk is below a critical threshold, so that a certain amount of remediation may be required, but it's not a place that you have to keep people out of. So now I'll just talk a little bit about how do organisms within food webs deal with toxic substances. Many organisms are able to detoxify. We are able to detoxify to some extent. We have various mechanisms that chemically transfer or sequester chemicals into certain less reactive forms within our bodies. And so this is a key aspect, especially when it comes to metals, especially microorganisms, which have a much greater diversity of metabolic processes that they can follow. They have various things that they can do, including making what we call organometallic compounds. We're going to talk about this a fair amount today. And a couple of things can happen with these organometallic compounds. Can be volatile and can be released. That's a key part of the mercury cycle. They can also allow a metal, because it's got these alkyl groups on it, to be stored away in fat. This is a way that, for instance, humans become exposed to toxic levels of mercury because there are some organisms, including food fish, that are able to recognize this detoxification mechanism. They just kind of store it away in their fat and it doesn't hurt them. We don't recognize this detoxification mechanism, and we eat the fish and it's got the mercury, we get exposed to it. And so we have to think about not only individual organisms detoxifying things, but what are the impacts on food webs, and how do chemicals get passed up through food webs? Are they toxic or not toxic? So I have some slides that kind of summarize some of these processes. There's a lot of them, but this is just a summary of a cell showing you a whole bunch of different things, most of which you know, we're familiar with. We've talked about oxidation and reduction, the precipitation of chemicals as salts, in this case, cadmium as a sulfide or a phosphate, but you can also find carbonates. You can find biosurfactant complexation is essentially the release of chemicals that coat surfaces, that trap materials. Extracellular polysaccharide sequestration is like the putting out of a mucus-like substance that again traps things. And there are actually some microbial populations that use this as a strategy to gather metals out of the environment, especially when we go down the redox ladder to do chemosynthesis from that. Here's this process. It says volatilization here, but you can see mercury has gone to methyl mercury, and we'll talk about that alkylation process. That chemical is relatively volatile. Intercellular sequestration, and this can take the form of solubility into fat or solubility into granules of solid materials. And in this very complicated sort of process, in this case using metallothionein, but there are other proteins that do this as well, that bind metals and keep them kind of irreversibly sequestered from the intercellular fluids. These are just some more additional processes, some of them the same as before, such as the polysaccharide, but also, you know, kind of highlighting that the role of passing things back and forth through the cell wall is kind of important, either through the lipid portion or the water-soluble portion. And so a lot of the detoxification mechanisms will promote kind of export in the cases where it can happen. So I'm just going to highlight one of these many things, which is microbial alkylation. We're going to think about it in sediments. It also works in soils. It's really dependent on being able to go down the redox ladder, because for a metal to accept a carbon as an alkyl group, it usually needs to be reduced. And so we need reducing environments. This is yet another reason why the redox ladder is so important. So this is a table of a huge number of metals that take a methyl group onto them, and the E0 of the transformation that is required. So we can think about, like, here's the, for instance, the PE or the E0 value for the iron 3 to iron 2 couple is 0.77. So these things here that happen at E0s above this value, yeah, we'll find those conditions pretty easily in the upper part of sediment columns because they're more oxidized than this. Where some of these over here, these require lower values, more reducing than iron. So we got to get down to usually sulfide, you know, production, or even methanogenesis, so deeper in sediment columns. But we do find them happening. And the distinction between these two things is kind of more mechanistic. It's a question of whether or not there's a methyl radical or a methyl minus that's transferred. And these are enzymatically driven reactions, so there's just different pathways in organisms. So these are just some examples. This is inorganic arsenic as arsenic acid. And you can see there's a series of steps here. This is a reduction, right? We're adding electrons, so we make it into the this tri-OH, and then a methyl group abbreviated as Me, that's a CH3, gets added, and then more reduction, and we get methyl group added back onto a structure that's kind of analogous to a carboxylic acid with arsenic in the middle. We've got now two methyls. There's still two methyls, more reduction, and then boom, now we've got three methyls on it. And this molecule is what's called an organometallic molecule, and it is much more soluble in fats, and it's relatively heavy because of arsenic, so it's not super volatile, but it's definitely more volatile than that would be. And that's something that happens at fairly reducing conditions. A lot of these alkylation processes are done by this kind of complex, this type. It's a kind of a modification of methionine. You need molecules that can have an alkyl group on them that they can transfer. So this group has a methyl group that it can transfer that's connected to a sulfur. This folic acid derivative has a methyl group that it can transfer relatively easy. There's something about this bond which is, you know, not that hard to break, so that methyl can be transferred over. This is a derivative of vitamin B12, and again, there's a cobalt in the center atom that can hold a methyl that is relatively easy to detach. And so lots of organisms use this mechanism. So this is a kind of complicated example for selenium. This is like within a cell, and this is the extracellular fluid in the environment. And so we start with some inorganic selenium. It's shown there in a couple of places. And it gets passed through the cell wall and reduced. Part of the critical aspect of this process is almost always a reduction. And then it's being transformed. And the transformation in this case that helps with this reduction and so forth, the combination of ATP and glutathione. And then you can see here that there's these methylated compounds, and these are going to be analogous to what we see with mercury, where we can have the methylated selenium. We can also have this sulfide compound, selenium bound, excuse me, this one here bound to a sulfur, bound to a methyl. And again, that's a very important form for mercury. And the way we got from this to this, you know, this is being excreted by the organism, is shown here. It's a complicated set of processes, enzymatically driven, lots of intermediates, oxidation, reduction, and then producing these things that can be fairly volatile, can be photodegraded in the environment back to forms that are more volatile and mobile, and ultimately reoxidize, kind of starting the process over again. So like I said, there's a complicated chemistry that happens in a combination of sometimes in the cell, in the sediment, at the sediment water interface, or in the water. And this kind of illustrates that from mercury, like I say, with some of those same forms. So we've got mercury in water, and it is transformed in the sediments here. You've got mercury +2. It can just be reduced by bacteria to mercury vapor, you know, mercury 0 valent, which is a vapor. It can go out. It can also be methylated, or this is that sulfide compound that I was talking about. Both of these are fairly volatile. This form is soluble in water. This dimethyl form is really volatile. It gets back up to the water column. And when stuff gets up into the water, in this case, this passes back up to the atmosphere and maybe redeposits. This is some kind of photochemical interaction. We'll talk about photochemistry later when we talk about the atmosphere. But interaction of energetic light can decompose this chemical and make it back into mercury vapor. But these compounds are something that can be bioaccumulated by other organisms. And this is the kind of primary way into which mercury gets its way into food webs, and that business about detoxifying and sequestering by organisms up the food web of this form keeps sort of protection to a lot of members of food webs. Unfortunately for us, we're not included in that category. And so these are just a couple more pictures of how this might look in different scenarios. So there's emissions of mercury, and you can see here that there are natural emissions. There's a small amount of mercury that comes out of volcanoes, for instance, and then local emissions, which are primarily not industries that use mercury. That was the olden days, like the Mad Hatter and this kind of thing, but you know, coal and fossil fuel burning, especially diesel, and to generate electricity, puts a small amount of mercury per unit of energy produced. But when you do it all the time over a long period of time, you have local impacts from that. And so that mercury gets into the environment, whether it's emitted as the 0 valent vapor or an oxidized form, it will oxidize in the atmosphere. And then it will go through all of those things that we talked about. You can see here, especially in anoxic environments that occur in sediments, the transformations, then released back up into the water column, and then into, you know, single cellular organisms, and then up into the food web. And yeah.
So the Hg2, is that like the
2+.
2+?
2+, yeah. And this is just another look at sort of how is essentially the same scenario, but showing you mercury emissions coming out of smokestacks, especially associated with coal-fired power plants and diesel-powered power plants, and places where the transformations take place. We really need anoxic environments. And so places where there are wetlands, for instance, or lakes, or shallow marine ecosystems right nearby. Just think like where Kahe Power Plant is located and what might be in the fallout zone. As long as we get into a reducing condition somewhere at depth where this is happening, then those transformations are taking place. Those transformations bring things back up into the food web. And an important part here that's shown here, which isn't indicated on this last slide, are bivalves, things that live at the sediment water interface that filter feed the water. When we have these chemicals percolating back up and they are filter feeding on that, they accumulate heavy metals. This is kind of one of the reasons that I don't eat any clams or oysters or anything that lives at the sediment water interface, because they naturally accumulate a lot of toxins out of that environment. Okay, so the next topic is to talk about how do we quantify toxicity. And I just love this graphic I found on the web. So it's just so intense. The first thing to think about is whether we're talking about metals or other compounds. Again, I'm going to focus on metals, but other kind of compounds, we can put them into a couple of categories. Some metals are essential, right? Iron and cobalt, and the list goes on, copper. And so in those cases, we look at the concentration of the metal, and we look at activity within an environment. This can be measured different ways, for instance, growth rates or enzymatic activity. And in this case, we're looking at growth. We'll typically find this kind of curve, and this curve may be different for different populations of the same species in different environments as a function of things like temperature and other environmental conditions. But the key point is that metal concentration is sort of an ideal concentration. And if you're below it, it's deficient. And if you're above it, then it's starting to have toxic effects. And critically, there's some concentration over which it's lethal, right? And again, it starts to get a little bit complicated because not all members of all populations will die out at a specific number. But what we're trying to understand is, what is this threshold? And what is this threshold? Now, there are other metals that, as far as we know, there is no essential function within the body, whether we're talking about a human or another organism. And the examples here of cadmium and lead are given, where there's a certain concentration where it's tolerable, right? It's not affecting the growth, but it's not needed. And then we reach a certain concentration where, again, it becomes toxic and then lethal. And so understanding the distinction between these two things is kind of critical. It's also important to recognize that oftentimes chemicals that are extracted from the environment for human uses, a good example is zinc, come along with these kinds of metals like cadmium. So most of the cadmium pollution on the planet comes from the mining of zinc. Okay, so another thing to think about are food chain factors, and how do contaminants, once introduced into the environment and once sort of absorbed or accumulated by the lowest members of a food web, how do those chemicals move up through the chain? And whereas lots of organic chemicals can be transformed by organisms, metals can't really be broken down. They can be transformed from one form to another, like a different oxidation state, or alkylated or dealkylated, which for that particular organism may help it sort of tolerate the existence, but they tend to not be broken down as easily, and in many cases not excreted, which means they build up and they move up through populations. And so just like kind of other chemicals in the environment, we can think about contaminants as being kind of conservative or non-conservative from the perspective of organisms, right? So if we have a acute exposure to a chemical that is water soluble, we get it, but we flush it back out. That means it's sort of non-conservative. Whereas if we get an acute exposure of a chemical that is fat soluble that we don't excrete, then that becomes conservative and it starts to build up over time. And this matters. So you can think about the food chain in a lot of different ways. This is just an example of a terrestrial food web. You could also obviously have a marine one for this example, but we need to think from the perspective of our contaminant, how it gets into the environment, and then all of these different kinds of interactions between the geosphere and the biosphere, the hydrosphere and the biosphere, and the atmosphere and the biosphere, and within ecosystem exchanges, things going up and down the food web, including import and export mechanisms, the production of fecal pellets, et cetera, et cetera. And so, whereas we might think of chemicals in groundwater, getting into soil, into the exchangeable fraction upon which grasses are growing, and animals are eating on that, and we're extracting these chemicals, these food stuffs, whether it's plants like corn or whatever that is, a cow, I guess, and then eating that stuff. This is a common way for contaminants to be introduced into foods that we interact with. And even if you take this part out of the equation, there can be very significant impacts in environments. Just think about the example I showed you of the zinc smelter a few weeks ago, where there was that kill zone in a forest that phytoremediation was being used to clean up. That has all sorts of impacts on the environment because contaminants are being used. So the two key factors, and these are oftentimes confused, and you'll see, like if you read a story in the newspaper, they will often call things bioaccumulation when they're not. They're actually a process of biomagnification. So I like to clarify what the two of them are. You can find all sorts of infographics on the web about this, but I put a couple in here that I think are pretty useful. What bioaccumulation is the concentration of a contaminant in the body of an organism over time because of exposure. And so you can see here the example of, here's a fish, and it's living in this water. These orange, red dots are supposed to be the contaminant. And you can see over time, the fish has developed a lot more contamination in their body. And that's a process of what we call bioaccumulation. And so that deals with members of individual populations at individual trophic levels of a food web. Biomagnification, which is the problem that, this is a problem, no doubt, and this is why they tell you, oh, if you're going to eat tuna, eat the young ones, eat the small ones, right? But this process of biomagnification, which is the increase of concentration of a contaminant as we move up trophic levels from, for instance, phytoplankton to zooplankton to small fish to big fish to seabirds, where the concentration of the contaminant increases at each level, is a much kind of worse problem, especially for human exposures. These are just a couple of other images showing you, for instance, bioaccumulation as these members of these populations age and get bigger, the amount of contaminant they have goes up. And whereas in biomagnification, we're looking at the concentration of contaminant increasing as we go up through the different trophic levels of a food web. So this is an example of how biomagnification looks. This is different locations in Finland, different places. And this is the concentration of mercury in food fish in parts per million, 0.51, and the concentration of mercury in the feathers of birds that are eating these fish, 0, 10, 20, 30. So you can see these concentrations are much higher than these. That's the biomagnification part. There's also this fairly linear relationship between higher concentration in the fish means higher concentration in the birds up to a certain point, right? Where presumably the concentration is high enough that the birds are not able to continue to accumulate without having toxic effects, and so we see turnover in that curve. There's a maximum concentration in the birds with higher concentrations are going. So we also have to think a little bit about the concept of what we call differentiation. When an organism is exposed to contaminants in the environment, and this is just an example of a contaminant, associated with rock particles in soil and living tissue with lichen, and we've got animals eating these materials. the accumulation of contaminants are not uniform throughout the body, right? And so, for instance, here's an example of, I'm not 100% certain if this is a seal, but I think it's a seal, and a moose with high concentrations of certain metals in certain tissues. So that, for instance, sometimes you'll see advisories that say, oh, don't eat the organ meat, of wild caught, whatever elk, because that's where these things are concentrated. And in fact, even though organ meat is very popular because there's also lots of vitamins and nutrients in them, you do have to be a little bit concerned about where it came from, because we do have the accumulation of a lot of metals there. And this is just an example showing you the amount of mercury in different tissues of a bivalve, something that lives at the sediment-water interface, where you can see in the muscle, the part that's typically eaten, concentration is actually relatively low, but in some of these other things like the shell and the gill and the viscera, the concentrations are much higher. So knowing something about the conditions in the organisms that are exposed can help prevent exposure to human populations by avoiding those materials. This is just a couple of other slides showing you some other aspects of contaminants in the environment. The idea of conservative versus non-conservative is reflected here. This is uptake of mercury by a bivalve. This is a control, this is time. So, in the control, there was no mercury. And then different concentrations of mercury at exposure, this bivalve and this bivalve, and then putting them back into fresh water that's uncontaminated and watching these organisms have concentrations that decrease again. So they're able to re-excrete. And in this case, this is a dynamic concentration that's supported by the accumulation from the environment, but it can be reversed for this particular organism. This is another case. This is an example of a diatom, which is a photosynthetic organism. They live in fresh water and in salt water. They make shells out of silicon. And so exposed to lead nitrate, with the idea of trying to see, whoa, how toxic is lead? And the reason this form was used is because it's one of the most soluble forms of lead. It turns out lead isn't particularly toxic to this organism. So if we look at time versus growth rate, these are the concentration of lead. The higher the concentration of lead, the better the growth rate of the organism. And in fact, what this means is that not that they like lead, is that they like nitrate. It's a nutrient. And so the lead just isn't all that toxic in that case. So now we get on to this quantification of toxicity. And this is the sort of gold standard that's used. It's not perfect, I'll say that right off the bat, but it's something called the LD50. LD stands for lethal dose. This is for acute exposure, something that happens, in a moment. And it is defined as the dose that an organism receives in a brief period of time over which 50% of a population dies from that exposure. Okay, and we can use this value when we look at LD values of different chemicals. They vary by many, many orders of magnitude, billions of times. Some things have LD50s with really low doses, and some have LD50s with very high doses, meaning it takes a lot to kill the members of the population. And this idea, marking it at 50% instead of 100% or some other value, helps to take into account all the vagaries of population dynamics, which we'll talk about, which can affect those concentrations. But it does bring up the point I want to, sort of express that when we look at LD50s, these are specifically for acute exposure, something that happens right away. In our world and in our environment, many of our exposures are something called chronic exposure, meaning continuous low-level exposure. The chemicals that we excrete again, then they have less of an impact usually than chemicals that we don't excrete that bioaccumulate in our bodies over time. But we measure toxicity slightly differently with this. With this, it's pretty straightforward. With this, what we're looking at is sort of average uptake over a long period of time that is sort of tolerable, or upper limit values. There isn't as much uniformity in the literature, like the US has what it uses, the European Union has what it uses, whereas for acute toxicity, everyone pretty much uses LD, which is lethal dose, LT, which is lethal time, and LC, which is lethal concentration. So this is just tables showing you some different chemicals and color coded by sort of things that are not at all toxic really, to things that are very toxic for both acute toxicity. And so you can see here we've been given an LD50 for humans in milligrams per kilogram. You see what the botulinum toxin is really low, and here's hydrogen cyanide. Even things like vitamin D3. If you take too much of it, it's toxic. To chemicals like whatever caffeine and vanilla and Roundup, whatever. Not that toxic. Still can have negative effects if you're exposed to a lot of it. So you can see the range of values here. And if you come over here to chronic toxicity, you'll see that these numbers for some of the same chemicals but exposed over time will have higher numbers of exposure. Lower numbers of exposure are of concern over significant doses. Now, the issues with this this system is that it assumes that all members of a population behave exactly the same in response to being in contact either chronically or acutely, and that just isn't the case. And so what we will actually find, this is a representation of the LD50 curve, is that in a population that has a normal distribution of population members of parameters that can affect sensitivity, and we'll look at them on another slide, we can find, the bulk of them are quote unquote normal or within a narrow range, but that can be hypersensitive members that can have toxic effects at much less than the lethal dose, where 50%, and we can have hyposensitive people. So, you can find people who say, oh, I've been smoking every day of my life and I don't have cancer and I don't have, any of the other toxic effects of nicotine. And it could be because they're hyposensitive, right? You can also have people that never smoke, but they happen to be near some secondhand smoke and they have toxic effects because they're hypersensitive. And so this is the reality of real populations. And obviously, when parameters are set to define what are the acceptable levels of contaminants in the environment, they're set at levels that are orders of magnitude less than the LD50. We don't even want to have an LD 0.001, right? But knowing what the LD value is gives us a set point to understand the relative toxicity of chemicals. There's some other things that are highlighted on this diagram which are important to note, which is that toxicity as defined by the LD is kind of like the ultimate toxicity, but what we're actually concerned with are sublethal effects, right? Things that don't kill you but still have impacts. Think about frogs with 3 legs or long-term, chronic changes in the human body from exposure. These are things that ideally you want to try and avoid as well. And one of the other aspects of this that's important is what happens when an organism is exposed to a toxicant. Is it reversible or irreversible, as per that example I showed you, the bivalves. So this is taken out of a textbook. I'm not going to try to go through this in detail, but explains to you how these measurements are made and the relative importance of not only the lethal dose, but the amount of time over which the exposure is. So, you can see here that oftentimes lethal concentrations, the amount of contaminant in an environment to sort of, if it's done in a lab, but to mimic exposure is over, a certain period of time, in this case, 4 days. That's a pretty common acute exposure. You can sometimes see acute exposures with different values. So when you see an LD50, actually, to be able to compare LD50s from different studies, you want to know, well, under what conditions were they made? What was the concentration used to achieve that dose? And how much time? And so we have the LT, which is the lethal time, LT50 of 96, with a 96-hour LC of, whatever the metal is that's involved, is, sort of how this is done. And you can kind of see here how, for instance, this is mortality in the for zinc in mussels, and these are different curves at different concentrations, different modalities shown here, and you can see that the LT is different. So you still achieve lethal dose toxicity in all 3 of these cases. Once you get down to this case, you're obviously moving up the curve much more slowly. This experiment wasn't run long enough to see if we achieve an LD along this curve or not. But this gives you an idea of the range of concentrations that would still cause an LD at different concentrations over different periods of time, if that makes sense. And this is just another infographic that I found recently in a paper. It's a really good review article. It's linked here. And I have a few images from this article that just talk a lot about the mechanisms of metal toxicity in the environment. And this is so color-coded for kind of like that other infographic that I showed you. Good, pretty good still, getting maybe a little bit not so good, less good, and then the red and the orange are like, yeah, really not good. And this is kind of a log scale. The scale bar is kind of coming up here in the middle. And so you can see here that as the concentration at the LD50 goes down, the more toxic it is, if that makes sense. The less of it you need to have. So this is just a zoom in on this part here to just kind of show you some of these molecules. So, right, potassium cyanide, mercury as an oxide, sarin, and then flip over here, you got ethyl alcohol, which is this one here, and N-hexane. Those things are, yeah, I mean, you could, if you ingest enough of it, they're toxic. If you ingest enough water, it's toxic. But the ones that are the most concerning in the environment are the ones with really low concentrations that achieve the LD50. So this is a table from your text. This is organic substances, just showing you the kinds of ranges, mostly organic substances. Obviously, table salt, and I've added in polonium-210, which is one of the most toxic materials. I added this in whenever, about 10 years ago, when some Russian former or current FSB agents killed a spy in the UK using polonium. I use polonium in my lab all the time, so it was of interest to me. That's a really small value. That's a big value, right? There's 10 orders of magnitude between these things. And there's some categories of, really toxic, sort of toxic, slightly toxic, and non-toxic, and that can be defined with those numbers. And this is from that same paper with that kind of spiral infographic showing you metals and their relative toxicity, again, with a color code system, and kind of highlighting that for many metals, it depends on what form they're in the environment. So that, for instance, when we look at metals in the oxide form or the chloride form, we'll see that in the oxide form, a lot of these are not toxic. They're less toxic, except for mercury. It's more toxic as the oxide, whereas the chloride form is more toxic for most elements. For mercury, we've gone from red down to yellow, but for most of the other elements, we've gone from green up to yellow. And there are other forms of these metals as well. This is just an example. This is further kind of zooming in on one chemical. This is chromium. Chromium in various forms. The oxide is the +6 form. The chloride is the +6 form. The chloride is the +2 form. Nitrate, etc. And you can see here different toxicology. These are done in rats, and so that's it's a pretty big difference. That's whatever that is, like a factor of 300, just based on the chemical. Okay, so remember at the very beginning, I introduced this idea of, well, we also want to know how do our metals get introduced into the environment. Are they particles? Are they dissolved in water, et cetera, et cetera. Think about all the stuff we've already talked about this semester of interactions between things that are in water with particle surfaces, oxidation reduction, organic matter complexation, Lewis acid, Lewis base interactions. And all of these things affect the form of the chemical, the bioavailability of the chemical, and therefore its toxicity. And I think you can recognize pretty much all the processes on here. This is just any metal with any charge, right? Binding to a clay, interacting with organisms, either on cell walls or out in the water column itself or in soils, being dissolved or not, interaction with humic acids. So these are all organic interactions, and down here are inorganic interactions, which involve complexation and chelation and oxidation reduction, et cetera, et cetera. And so where a metal may be introduced into an environment in a place where it's oxidized, and if we move down gradient, for instance, groundwater flow, where it becomes non-oxidized, that may change the behavior. Now I want to talk a little bit about what we call non-lethal or sublethal toxic effects. So this is an example of copper exposure, in time and days, and the number of survivors. And so you can see here different concentrations, right? This is a really high concentration, a little bit lower, a little bit lower, a little bit lower, about 10 times lower. And so you can see that the length of time it takes to reach the LD50 or the LT50 in this case gets longer and longer and longer when we get to lower concentrations, but we still achieve it at a value of 0.4 for these organisms, but a value of 0.3, it doesn't seem to. So that doesn't mean that we're out of the woods, that those, it's true that we're not killing the organisms at that concentration, but they're probably almost certainly what we call sublethal toxic effects. And this is an example of sort of normal larvae and an impacted larva for organisms that are, seeing high concentrations of copper or zinc. And there's myriad numbers of these examples, so that, even values of 0.1, while the LT50 might be infinite relative to the lifespan of that organism, that doesn't mean that there aren't impacts. And that's why we want to use sublethal effects when we know them to quantify toxicity. Okay, so now we'll talk a little bit about how is toxicity manifested in organisms. And this is just an example of some of the ways that, for instance, metals can impact metabolic function. They can inhibit enzymes, by binding irreversibly. They can inhibit protein synthesis. They can affect cross-membrane kind of distribution of other chemicals, and they can target certain specific organisms and affect organs and affect how those organs function. And this is just a diagram showing you some of those things for the human body. And again, some of the different ingestion mechanisms, right? So we talked about ingestion, inhalation, dermal contact, for instance, and the pathways through which that stuff comes into the body by one of those mechanisms will impact different organs, lungs, heart, whatever. They're all related together. And so toxicity mechanisms are important for understanding how populations are exposed and how the effects can be reversed, or if they can be reversed. But they don't matter so much in terms of defining what is the absolute limit that we want to set for exposure. So this is just another diagram that shows you some of the very specific toxic effects, most common, doesn't mean it's the only one, but the most common for lead, mercury, and cadmium, and copper. And you can see that there are kind of a couple of categories of effects: DNA damage, for instance, and impacts to cell division and embryonic effects. You'll also see that you'll see a lot of MTs, and so MT is metallothionein. It's a really important chemical that we and many other organisms use for sequestering metals to a certain point, but we can saturate it. We don't have that much of it in our body. We don't produce that much. And so, high chronic exposure or even acute exposure to metals can disrupt that metallothionein effectiveness. So there's a lot of other factors that are related to kind of overall toxicity of the elements. This is another diagram that sort of shows kind of three categories of elements. Elements that, they're not that toxic in terms of the LD50 values. Elements that are toxic but are rare in the environment and or very insoluble, so not particularly mobile, and elements that are very toxic and relatively accessible because of mobility. So this is just sort of a generic look at, it doesn't tell us which chemical form or anything else, but what's interesting is when you look at this and you look at the periodic chart, you'll start to see that a lot of these guys are relatively high electronegativity, or they have high affinity for sulfur or high affinity for chlorine, and or some, to a lesser extent, some of the other Lewis bases that we find that are oxygen bearing and nitrogen bearing, and that's what affects their ability to disrupt systems within organisms and therefore be toxic. So here's some of the other things to consider about heavy metals in the environment, the sort of toxicological properties, which are like, well, how persistent are they in an environment, like in the soil zone, if that's what we're thinking about, or in the upper water column, if that's what we're thinking about. Can they be transformed and excreted or removed from water by precipitation or something like that? You know, what's their residence time? What's the acute toxicity, meaning the LD50 for various members of food webs? What kinds of bioaccumulation, biomagnification factors? What are the chronic sublethal effects? Synergistic effects are when one chemical along with another chemical together work in concert, either to counteract each other or to reinforce each other, or things that like behavioral things, like for instance, small amounts of certain chemicals, which for instance, we can detect with our smell, may cause us to avoid areas that have other kinds of toxicity in them. So there's a whole bunch of different kinds of things. And then these sort of tetogenic and carcinogenic effects. So we've talked about most of these things already, but I just kind of want to highlight that those are some similar aspects. And this is a rather complicated, it's actually two tables from the same textbook that I put together to try and reinforce most of the things that we think about with respect to contaminants in the environment. So there's all this stuff here, which we've been talking about all semester, like what is the form of the metal in water? How does, inorganic, organic, soluble, particulate, complexation, chelation, colloidal, precipitated, sorb, all that stuff. And that's what this table comes in, right, with the different size fractions and the different complexing agents and chelating agents and particulates, etc. But then we come to these other factors here, which influence things like population dynamics. So are there synergistic effects with multiple metals impacting them? How do other factors in the environment affect populations? For instance, temperature, pH, or dissolved oxygen, or light, or salinity for aquatic organisms, this was from an aquatic pollution book, so everything focuses on aquatic stuff. So that if organisms are living in their ideal range where they're thriving with respect to each of these parameters, then the LD50 value for toxicants might be higher than if they're living at the edge of their ideal range, because they're already biologically stressed for other reasons. And then we've got all these conditions of the organism, stage in life history, right, age, larva, etc., changes in life cycle, age and size, sex, starvation, activity, additional protection, physical protection like the shell, adaptation to metals, altered behaviors, etc., etc. These are the kinds of parameters that make the LD50 curve look like it does, and to have hyper and hyposensitive members of a population. So it could be, for instance, the females or the males of a species are more susceptible to a particular contaminant because of biological differences. And that manifests itself in not only in the LD values, but also in setting acceptable limits that are well below the LDs for exposure. So for the last part today, I just have a little bit of stuff about kind of how do pollutants get into the environment. This is obviously a really brief summary, but just sort of to think a little bit about that. So this is a table of some, pretty significant contaminants, all of which have pretty low values of LD50 in certain forms: arsenic, cadmium, chromium, lead, mercury, selenium, silver. What are the forms that we typically see in water, and what are the natural mineral sources? And the reason that's important is that most of these metals are used in one or another industrial process, and the way that we gather these things as a sort of a civilization is by mining them out of the environment, right? And that would come from mineral forms. And so you can see, sort of arsenates and sulfides and oxides, in a couple of cases, chlorides. But these are things that are taken out of the environment, and you can see what they're used for. A lot of arsenic has been put into the environment. We'll have a focus on arsenic coming up in a later lecture, where it's used in lots of agricultural processes, for instance, and treatment of wood. So there's a lot of arsenic, even though it's no longer sort of acceptable in a lot of countries, a lot of legacy arsenic out there. And each of these metals has various sources or uses in automotive industry. You can see ammunition, you can see dyes, electroplating, electronics, et cetera, et cetera. So they're really pervasive. And we throw a lot of this stuff out, so it goes into our landfills as well. So this is another way of looking at the sources of pollutants from different processes. So there's the process of mining and refining of ore, smelting in the metallurgical industries, and they do introduce a lot. But then we've got stuff like corrosion of metals. Just think about rusty things that are out in your yard that are using metal alloys. They may have small amounts of heavy metals mixed in with iron. Waste disposal, obviously, fossil fuel combustion, agriculture and forestry. All these things are sources of metals to the environment as modern practices dictate. And this is sort of another table showing you kind of the bunch of metals, some similar to two slides ago, what they're used to. The reason I put it in here is this percent recycled. And these numbers are obviously not high enough. If the recycling rate is higher, then you're introducing less into the environment, reintroducing, and you're having to do less of the recovery of new mineral ore and the smelting and the refining. So there's multiple points at which you release less chemical into the environment. And in fact, for many industries, it saved money if there was more recycling. It's complicated because in a lot of these cases, these are distributed metals. There's small amounts of them. So if you think about your cell phone, how much of which of these metals are in there. There's definitely copper, there's gold, there's a little bit of lead, there may be a little bit of mercury, there's definitely silver in them. You collect all those cell phones, who knows where they go when you upgrade to your next phone. And I always like to imagine that they're giving them to some people who want my old phone, but probably not. They're probably just getting crushed and thrown out. And yet if they were re-extracting those metals, I think that integrated over, a billion phones, it starts to matter a lot. So this is a box model, kind of a quantitative model of an environment where heavy metals are being introduced. This happens to be the North Sea. And these are things that we've started to talk about already throughout the semester. But if you want to think about, all right, we got metals in this environment, and how are they getting there? What is their fate in transport? And how are organisms being exposed to metals? We have to think about a complicated flow chart like this, right? Where we could be looking in the soil in a nearshore region, or we could be looking in the water of the estuary, or the nearshore zone, or fresh water, small lakes or rivers. We could be looking at the air. But these are all the kinds of exchanges and transformations. We've talked about most of these things already. What causes a particular metal to be flowing down a river and decide to partition itself between the dissolved form and the form that is dumped into sediments and different metals, transition through the estuary and mixing zone between saltwater and freshwater into the oceans. Some of them increase in concentration, some decrease in concentration, depending on various other chemicals that are in the environment and so forth. And so this is just a box model showing you all the possible things that could happen, superimposed upon by metal fluxes into the environment. So for instance, incineration, smelting, and exhaust goes into the air. Air produces fallout, which can then, the fallout arrow interacts with every one of these systems. For instance, dumping of materials might more affect the water, which then it introduces into all of these systems. Dredging, which is, I think I mentioned this when we talked about estuaries. A lot of estuaries are places with population centers because of the sort of developing over time because of the rich biological life there. And oftentimes these became places where harbors were established. And so all the processes that go on in an estuary, and also you put ships in there, right? And ships do all sorts of stuff, not on purpose necessarily, but they've got paints on them that are leaching metals into the water so that things don't grow on them, and they're allowing fuel oil, which has metal contaminants in it, into the environment, and they're, other processes as well that happen sort of onshore at harbors introduce a lot of metals into this very dynamic environment. And then every once in a while, harbors are dredged, those really nasty sediments that even if they were just estuarine sediments without a harbor, would already be rich in heavy metals. They become very, very rich in heavy metals, depending on where this stuff gets put, right? Some places that gets put into a landfill, some places it gets barged out and dropped into the shallow sea. It doesn't mean it's gone, it just means it's transferred to a different part of the system. And depending on, for instance, where the dredged spoils are put, if they're put on the surface of the seabed, that salting off process, which you talked about, can affect some metals, some lead, cadmium, zinc. They can come off those metals back into the water column and be introduced back into the ecosystems that interact with each one of these different parts of the environment. So there's a lot of different fates of metals in the environment that we have to think about, like what is their solubility as a function of environmental conditions, like temperature, how do they behave as a function of other parameters of water chemistry. Here is given an example of hard water. So if there's calcium carbonate precipitants, what does that do to the concentration? Some metals like zinc will what we call cement out. They will be removed by the calcium carbonate and so reduced in concentration. What's the acidity of the water? How does that affect solubility? How are insoluble metals affected by sediment fluxes in an environment? What is the interaction with aquatic organisms, all the food chain factors that we've talked about? And what about the benthos, the organisms living at the kind of water sediment interface or deep in the water column? How do they interact with chemicals? There are other things too, that's just a few of them. And so this is just a little bit of information that goes along with sort of this model for heavy metals, copper, mercury, lead, and zinc, and the rivers, the dredgings, the atmosphere, the direct discharge, industrial dumping, the sewage. I'm going to show you on the next slide something related to this. But you can see for these metals that it turns out that the two highest concentrations into the North Sea come in rivers, both soluble and particulate load, and atmospheric deposition. And this is impacted dramatically by human activities, especially industrial activities, which release vapors into the atmosphere, which then resettle out, which is something that can be addressed by sort of end of pipe measures to help reduce those discharges. And some of these other things, they're important, not to minimize them, but direct discharge, industrial dumping, sewage sludge, and incineration, they're smaller, in some cases a lot smaller. Direct discharge can be significant for some of these metals, as can dredging. We get to the atmosphere, so that's right, it's highlighting the air in that last slide. This is a diagram showing you for a bunch of different metals, the anthropogenic source versus the natural source. So the natural source of heavy metals in the atmosphere is primarily volcanic. Another place where we can get some metals into the atmosphere during forest fires, especially huge fires. There's enough heat, there can be relatively small concentrations of these metals in natural unperturbed ecosystems that get distributed into the atmosphere. And so understanding what that baseline flux of metals is a little bit complicated in the modern world that's been so perturbed by human activities. But you can see here what these numbers are like. Total emissions, and this is a table from a paper in the 80s, a very influential paper. It hasn't been updated very much. I'm sure that most of these anthropogenic sources have gone up to some extent, but the key thing is that in a lot of environments, the amount of metal that gets introduced into the environment by natural processes is almost like half. So this is not to say that, oh, well, it's natural, let's go ahead and contaminate. What it means is that if the amount of natural flux is high enough to push an environment close to the thresholds that we're concerned from an LD perspective, then we can add less to the environment, right? It's kind of like, I remember the first time my doctor told me, you got high cholesterol. I go, oh, well, it runs in my family. It's biological. I don't care where it comes from. You got to limit your cholesterol. It's just harder for you because you have whatever genetic factors. And it's the same thing here. If you are downwind of Kilauea, where there's high concentrations of mercury and lead and cadmium and arsenic coming out of the volcanic gases, then that would be a bad place to introduce, sort of human practices that are also introducing those things into the environment. And so I like to emphasize this concept just so that you are aware that there are natural sources of materials in the environment. So this is an example of transfer of metals from, I just have a couple of slides, from the atmosphere to the sea surface. These are in nanograms per centimeter squared per year, centimeter squared of land area, nanograms being 10 to the minus 9 grams, so a billionth of a gram, or a millionth of a milligram. These are pretty small concentrations. And you can see that depending on where you are, you've got different concentrations of all of these metals. And here we are in the tropical North Pacific. We're in the best case scenario because we're the farthest from both the natural and the anthropogenic sources, right? And the fluxes are significant. If we look at, in the Mediterranean and the North Sea, all these concentrations are higher. Like this is 10 times higher, and this is, 50 times higher. So it does matter from an from a chronic exposure perspective where you are on the planet. Oh, I forgot. I guess that's the last one. I thought I had one other related to this, but anyway, that's toxicity kind of in a nutshell. And like I say, after break, we'll come and we'll look at some specific heavy metals and some specific environments in more detail, kind of keeping all this stuff in consideration. So I want to say this, so it's on the video. I am postponing the midterm until after break. You will get it after break. Don't worry about it. I've got several emails. I will alert you ahead of time. It's something that you'll have a week to work on. I think it's going to take a couple hours, something like that, maybe 3 hours. It's going to be open book, open note, use the internet, use whatever you want. Don't work together. And I'm just going to give you some news articles on different topics and then ask you some questions about it. That's basically how the exam is. I think the problems are easier than homework. Questions? Any questions?
That's going to be uploaded to La Lima, or?
Yeah, and I will send, like for the midterm, I will definitely send a message,
Okay.
Saying, yes, the midterm has been assigned, but I'm going to do it, like I say, after break. So I guess the Monday or Tuesday after break.
Oh, okay.
Yeah. So I don't want you to have to worry about it.
I have some friends in town.